It looks like we have a few people trickling in, but we're also 4 minutes past the hour, so I'm gonna go ahead and kick this off. So welcome everyone to this IRIS-HEP Topical meeting, and today we're joined by Dr. Titus Brown at UC Davis, who is going to be talking to us about workflow language in bioinformatics and how he and his team are able to use these to do analysis at the petabyte scale. And then also maybe how there could be some use cases beyond bioinformatics as well. So without any further ado, Titus, please take it away.
All right. Great. Thank you for the invitation. I'm going to be talking about workflow languages in bioinformatics. As Matthew said, I'm at UC Davis. I'm actually in the School of Veterinary Medicine, where I run the lab for data-intensive biology. And feel free to contact me, however, by hook or by crook. These days, I'm hanging out on Blue Sky a fair bit, which is a newish social media platform. And I'm accessible via email as well. A couple intro warnings. So this talk is mostly going to be about SnakeMake. This wasn't really an intentional bait and switch. I didn't really remember the title when I was preparing my outline and slides, and then I looked at it this morning. I was like, oh, that's the title I gave. So I hope it'll be interesting. If not, I only have about 20 to 30 minutes of content. And if there aren't a lot of questions, then that's totally fine by me. But nobody ever gets too upset by too short a talk, as far as I can tell. I do have lots of strong opinions, but they're pretty loosely held, and I'm hoping for discussion. So please feel free to ask for clarification, post questions, raise your hand, however however you want to do it. And I have the chat up on my screen, so you can just I might not see the raised hands because I don't think I have the. Let's see, I don't think I have that. Can't see the participant screen at the moment. Okay. So I came to biology. I just want to introduce myself a little bit. I came to biology through physics, sort of, which is to say I have always worked, well, with one exception, all of my research work has been in biology or biology adjacent fields. That one exception is physical meteorology, where I worked on a project called Earthshine as an undergraduate. And Earthshine is sort of an astronomy project where we were using the moon as a mirror for sunlight reflecting off the Earth to assess the reflectivity of an entire hemisphere of the Earth at one time. That project's still ongoing. But the important thing is that all of my early training was actually by physicists. And so I come to biology sort of from that style of thinking and style of thought, but I've become a card-carrying biologist, or I guess my formal training, I was in biology. So I've done some modeling, a lot of simulation, an awful lot of data analysis, and a tremendous amount of genomics. I focus on large-scale data reuse. In a lot of my research, the sort of theory there that I think should fly well with this group is that often there's a lot of published data that hasn't really been explored in great detail. And this is, I feel like it's particularly true of biology. And so figuring out how to use and reuse existing data in tandem, perhaps with new data or just to ask new questions is something I've spent a lot of time on. At a sort of more philosophical level, I'm really interested in helping people extend their personal reach and also build capacity on a sort of field-wide scale. I feel like there's a big gap in biology between what we could be doing if we had more computational capability in the field among if we trained people better, and what we actually currently have at the moment. And so I work a lot. I think a lot about training and community and development of resources. I'm very focused on open source, open science, reproducibility. A lot of my introductory work before research was actually open source software development. And I brought that to all of my research now that I'm sort of an old geezer as a professor at UC Davis. I still think a lot about these things. And then my lab and I both do we all do a fair bit of software development engineering, and you can check out our GitHub repositories there. The first one's my personal one, and the latter one is my lab's. So, I'm guessing if you showed up to this talk, you probably aren't going, "What's a workflow system? Why would I need a workflow system?" And so this is really a personal introduction to why workflows, which is just to say, over the years, I've realized that I've used workflow systems to address a set of paranoia I have about computers. And these are the three things where I go that I seem to be most paranoid about. One is that I never know. I always worry about jobs failing silently. I always worry about, missing an error code. I think this is also probably true in a lot of other fields, but in bioinformatics, you get pages and pages and pages of output from each job, status notes and so on and so forth. And very rarely are errors, are non-fatal errors, or even fatal errors called out very clearly. Often there's some janky script that's running something and it has a non-zero exit code, and you don't notice if you're just running in a shell script. And so workflow systems let me know when a job fails. You might think there are many other approaches that do that, and there are, but it's built like you can't, it's hard to turn it off in workflow systems, whereas in other systems you have to turn it on in shell scripts, for example. Workflow systems also let me pick up execution immediately after a previous failure. So if I'm running a whole bunch of jobs and I'm running 100,000 jobs and 50,000 of them complete, I can pick up at the 50,001st job once I fix whatever the error was, whether it's a transient error or a programming error or a data error or something else. And that way I don't have to unnecessarily repeat work. And then workflow systems manage concurrency. This is just, there are lots of ways to run things in parallel. And at least in my world, there's a lot of reasonably complicated direct acyclic graphs of sort of workflow of job dependencies. And I just can't be bothered. So to sort all that out and run things in the right order. And so I just let the workflow systems manage the concurrency for me. But if I had to take a step back and tell you what I really worry about, it's really the first one. I just really worry about having things fail and not really noticing, and then publishing the wrong, publishing something that is readily catchable. You'll note on here that automation is not actually on this list, and that's like I automate everything all the time, everywhere, with scripts and with various kinds of scripts and so on. And workflow systems are great for that, but I take automation as a given. When you're working at any large scale, you have to automate, and so these are really the things on top of automation that lead me to pick up workflow systems like SnakeMake over other options. The way that I think about workflow systems in the sort of overall space of software that I use, this is actually a slide from a 2016, I think, talk I gave in Oslo, or tutorial I gave in Oslo, Norway, is, I have some raw data. I want to take that raw data, generate some results. And there's some execution environment that I'm running everything in. And that's running, that can be HPC, virtual environment, virtual machine, cloud, Docker container, whatever it is. I don't really, none of my tools really care anymore as to how this execution environment, what exactly this execution environment is. It can be chosen by platform, but all of my tools work within any one of these execution environments. And then I use some kind of workflow engine, SnakeMake, PyDoit, whatever, to take the raw data, distill it down to usually sort of spreadsheet-sized results. And then I use those spreadsheet type results, those CSV files, or boiled down results. I feed them into a data analysis narrative, usually something like Jupyter Notebook in my case, or R Markdown or Shiny or something else. And that gives me the results that end up going to my talks, my discussions, and my papers. And of course, underneath all of this there's version control. So everything that I do, basically everything that I do is saved into some kind of version control system, usually posted to GitHub publicly or privately. So I think this is probably pretty standard again, but I just wanted to say my workflow engine is the first thing I run on the raw data That's usually the large stuff that takes days to weeks to run. And then once I get to the data analysis narrative, now it's a little more interactive. So as I alluded to at the beginning, SnakeMake is my workflow language of choice. And why? Why is that? Well, so there are hundreds of workflow systems, and it's not easy to figure out how to pick one. I've had many conversations over the years with people who insist that their the workflow system they use is the one that I should pick. And of course, when there's hundreds, like that's not everybody has their own favorite workflow system. And so the guideline, the reason I've picked SnakeMake is this: choosing a workflow system is really a long-term decision. You, your collaborators, other people in the community who try to use your code will probably be stuck with this decision for a while. So it's good to pick one that has legs, that has a community, and whose community overlaps with your domain, because that probably means that there's going to be documentation, there's going to be help available, there's going to be plugins or code that directly speaks to your use cases. And when you stop working on a package or on a workflow, and somebody else in your lab or in your community needs to pick it up, there's going to be other people there's going to be some basic knowledge in there of how to deal with that workflow, with the workflow language. And so in bioinformatics, the choices really boil down to SnakeMake, Nextflow, CWL, and WDL. I'll expand those acronyms a little bit. CWL is Common Workflow Language. WDL is Workflow Definition Language. CWL is a sort of community-based one. And WDL is something that I think primarily came out of the Broad Institute, which is one of the large genomics institutes out there. And I will just say my lab converged on SnakeMake through a nonlinear process. I had several people that used different workflow systems. I used Make for a long time. I still use Make for simple things. And then I had a particularly computationally capable person who did the sneaky thing of not just using SnakeMake, but teaching the biologists in the lab to use SnakeMake. And then I woke up one day and everybody in my lab was using SnakeMake. That's how my lab often converges on things, is I wake up one day and I'm like, oh, this decision has already been made. Let me recognize that and start learning this myself. At the time, I didn't know SnakeMake. So it was an interesting experience. So I think I'm speaking to mostly a physics crowd, and I'll say, so I wanted to include this slide. How different is bioinformatics from physics? And I don't really know. I don't really work in physics. I'll just say, in my experience, bioinformatics has more situations where you have many large files. It has more situations where you have many big RAM jobs, big memory jobs applied to those large files. This is in part because of just the underlying algorithms we use, but also due to some of the way that our software is written. We have much less numerical, but in the way of numerical processing and what some former colleagues trained me to use to call pleasantly parallel multi-threaded work. So there are some things that are multi-threaded. They tend to be very I/O dependent. So your threading situations care less about your processor architecture and more about can my file system handle what I'm about to do to it? But it's still, we still have some of those, but by and large, a lot of what we do is sort of single-threaded, somewhat big memory, somewhat data intensive, and you're doing it to hundreds or thousands of files. There's lots of format interconversions, lots of sorting. I think this is a generally accepted thing in bioinformatics. We have tons of just terrible formats, or really we have several just bad formats. They're all text-based. There's not much by way of random indexing or binary formats. It's kind of terrible. So an analogy that I put together about 10 years ago never really landed with the biologists, but in bioinformatics is kind of like everybody has their own personal particle accelerator for doing experiments and producing data. So there's not like there's a central place where large scale data is produced and then a common set of tools for filtering it. You instead sort of have this Wild West situation where every grad student, every postdoc has to sort of figure out their own processing pipeline for their own large scale data sets. And this is an interesting challenge sociologically, sociotechnically. So my summary here is basically with lots of medium and small sort of shell script jobs with lots of intermediate output files. And so, some things like MPI are basically not used at all in bioinformatics. SnakeMake, I don't know how many of you are familiar with SnakeMake, but in 5 slides or less, here's a basic SnakeMake rule. SnakeMake is basically a simple domain-specific language for workflows. Each rule must have a shell or a run command that tells you what to run, and then you have optional input and output blocks that tell you what this rule requires in order to run, and when this rule finishes successfully, what output files it will have created. And here you can see I'm using templating in the shell command to not repeat the file names. I can just take them from the input and output blocks. And so, of course, any SnakeMake file will generally consist of multiple rules like this, and the rules get changed. So if you say, I want the output file from this rule, and the input file for that rule doesn't exist, it will go see if there's another rule that has that provides that input file as that other rule's output file. So you get sort of automatic construction of directed acyclic graphs from sort of individual rule, declarative rule specification. SnakeMake has pretty robust templating. So here's a really simple example. I define a Python list that has 4 entries in this case, and I define a rule, and this is a rule that produces 4 files, and the 4 files are named, each of these accessions .fna.gz.sig, and it just fills in automatically, so you can update this and it'll fill in. This is what did I want to say about this? This is a nice example in SnakeMake. SnakeMake is built on top of Python. It in fact uses Python, the Python parser, and defines new meta class approaches to parse the rules. So you can introduce Python natively in your Snake file for manipulating strings and for input and output functions and other such things. SnakeMake also has robust wildcarding. So here I can say I have this rule all where I say build me all of these files that come where the files are named for a list of things that I've read in from a text file. So this text file could have 50,000 things in it. And then, except that I'm printing them, so you probably don't want to put 50,000 things in there, but maybe a couple dozen. And then in the rule all, you say this is the first rule. It's the rule that we run by default. The name doesn't particularly matter. When you run it, it's going to say, okay, build me all of these files. And then SnakeMake will look through the rest of the Snake file and go, okay, I have a declarative rule that tells me if I want to build a file named something.sig, then I can build it as long as this other file, genome/something.fna.gz exists. And if that does exist, I can take this input and produce this output by running this command, which is now a shell command that says, run this program sour mash with these parameters, take this input file, take each input file and produce this output file. So this is a nice example here, a nice simple example here. Sketch genome is a wildcard rule. So if you feed rule all, say 5,000 accessions, 5,000 different versions of sketch genome will be run, one for each value of accession, and these will be run in parallel if you give SnakeMake sufficient compute resources, or it will be run them, it'll run them in succession. So this is a wildcard rule. This is what I call a concrete rule in the sense that it's asking for a specific set of files. And then other rules in the Snake file are explaining how to make each one of those files. As I said, SnakeMake has good Python integration. You can, run these commands and treat all the results as if they're just Python variables and print them and so on and so forth and manipulate them. The only real challenge is that if you put Python code in here in the rule, you have to format it in a particular way so that it runs as part of the rule. Basically, the way this works is the way SnakeMake works is it reads the entire Snake file in, executing all the code at the module level, bef defines all the rules, and then it looks at the rule at the directed acyclic graph. It constructs a directed acyclic graph from the rules, and then decides what to run. And so you can't sort of naively put Python functions in random places in here and have it work. And I'm happy to talk about that a little bit more or give some examples. One of the best uses of SnakeMake, in my view, is you can apply operations to many files at once. Literally, if I just want to build a subset of a bunch of subsets from files, from files. So if I have like 100 different sequencing files and I want to just pull out the first 400 lines from each one, there's reasons why you want to do this sometimes. Literally, I can say, hey, pull in all the file names, load in all the file names that end with .fastq, pull out the names, build this set of these are the files that I want using all the names that I just learned. And then here's a rule to go from the whole file to the subset file. So I do this for file renaming. I do this for file subsetting. I do this for any sort of simple operation that I need to apply to dozens or hundreds of files. I'll use this over a for loop in Bash, for example, or something like that. So that's like a very brief introduction to sort of SnakeMake syntax and rules. And I'll say there are some things about SnakeMake that I don't love. It can be challenging to figure out how to build, how to update the directed acyclic graph of dependencies on the basis of previous outputs. So if, for example, you have a rule that produces a set of files that are unknown number, for example, a clustering rule, a rule a job that runs clustering on subset of outputs, you don't know how many clusters you're going to get, so you can't actually tell SnakeMake in advance what to do. So you have to dynamically inspect the output. SnakeMake lets you dynamically inspect the output of specifically designated rules and then construct sort of new rules based on that. And I think it's well implemented, but I haven't yet wrapped my head around it, and I've been trying for a while. So it's hard. It's confusing. I think that because another problem is that because they don't define their own parser, because SnakeMake doesn't define its own parser, when you define rules like this, if you have a syntax error in there, you end up at the mercy of Python's default parsing, which is sort of meant for programmers and not for data scientists who may not be particularly good Python programmers or particularly experienced Python programmers. So the error messages that SnakeMake outputs from parsing fails are often confusing. And I would say that's a general rule, and this is one of my hot takes. Like, SnakeMake is not super welcoming to newbies. Like, if you know Python and you know what you need to do at the command line, you can work your way through the initial examples and achieve success. But if you don't know how to program already, which is a situation that a lot of biologists and bioinformaticians are in when they're getting started, SnakeMake can be hard to learn. And then just a personal experience from my lab, SnakeMake has stopped working well past 50,000 jobs. We tend to, in the past, that may have been fixed, but once your directed acyclic graph gets complicated, big enough and complicated enough, we've had situations where SnakeMake has not operated with full efficiency. And I sort of want to mention that just in case there are people who are like, ooh, SnakeMake looks really cool. Let me try it on my 1 million file situation. There are various ways of dealing with this, but it goes above and beyond sort of the, oh, let me write a simple snake file. So I'm going to stop after this slide, after these next two slides, and see if there are any questions, because I know I've sort of been going at a good clip. So how does SnakeMake compare or contrast to other workflow languages in bioinformatics? Nextflow is great. I've heard great things about it. I just haven't used it. It has a great toolchain ecosystem. It has wonderful pre-built and reusable workflows. It's got a fantastic community. I think Nextflow and SnakeMake fill a different niche than these bigger. I would think of them as bigger, but maybe just different approaches that are the common workflow language and workflow definition languages. These are situations, CWL and WDL are standards, where a standard language for defining a workflow has been created. And then you can support multiple runners that work in different environments with different kinds of efficiency. And my sense is that these are often used by production platforms where you have a workflow that's already well understood and you just need to run it as efficiently as possible. And it's not where the niche that I think SnakeMaking and Nextflow work in, where you need to tweak the workflow pretty regularly for your own research needs. And I think CWO and WDL are an increasingly mature ecosystem that's worth investigating if you care about, just getting your data analyzed with a workflow that's well defined and it doesn't need to be super flexible. Something that we have explored is wrapping SnakeMake workflows in CWL or WDL. You can imagine a situation where, well, a situation we were in is that we wanted to take advantage of WDL's, WDL's advanced scheduling approaches to dealing with, and the cloud for dealing with IO intensive versus memory intensive stuff. Because in the cloud, like you want to spin up different machines, you want to run jobs on different machines. Probably in sufficiently complex HPC environments, you want to do the same thing. And WDL handles that really well. SnakeMake it might, but it seems more complicated. So we explored the idea of having a single SnakeMake workflow with multiple targets. And then we had in WDL, we ran the different targets in differently annotated bins. So we said these targets, this set of targets is IO intensive. Please run these on that kind of machine. And then this set of targets is big mem intensive. So please run that over there. And then each SnakeMake target actually consisted of maybe a dozen different commands that were running all in the same Docker container in WDL. I haven't decided if this is a good idea yet. We just did it once. It seemed to work great. So I'm not sure if I can recommend it, but it's something you certainly do. So last thought before I just pause for questions. Community considerations is really code word, partly a code word for being able to Google stuff. Stack Overflow, tool documentation, and ChatGPT are really common ways now that a lot of students and researchers are finding answers on the internet. Stack Overflow and tool docs have been around, obviously, for decades. Something that I'm starting to see students do a lot more is go to ChatGPT and say, hey, write me a SnakeMake workflow that does X, Y, or Z. And I think we can argue about whether that's a good idea or not, but it is being done. And so I'm in this interesting situation now where I'm starting to write my tool documentation and my examples with the knowledge that they are unlikely to be read by people, but are more likely to be digested by large language models that serve them up to people. Anyway, I'll just say, like, there is an existing community for a workflow system, then you're likely to be able to find answers. And I think probably everybody with sufficient computing experience has run into this problem where they get some weird error message. They post about it, they forget about it. 2 years later, 3 years later, they come across the same error message. It's never been answered. And you Google it, and the top hit on the internet is your own unanswered post from 2 years ago. And I hope that, if you have a sufficiently large community, there will be somebody who's like, oh, I ran into that too, and here's how I solved it. So being able to find answers on the internet is reasonably synonymous with some kind of community, at least in biology or bioinformatics. And I would just say SnakeMake and Nextflow have really robust online communities. It's probably worth considering fixing or extending SnakeMake to meet your needs versus writing your own workflow system. And I should just a shout out here, I think SnakeMake version 8 will have a robust plugin architecture, so you can add your own runners and perhaps even your own syntax in certain places. Not syntax, but your own semantics in certain places. So I'm going to just pause for 30 seconds to see if anybody wants to come forward with some questions. And again, I can't see raised hands, so please feel free to just drop something in chat or unmute and say something.
Yeah, I can see the participant list, so I can also help field these. Okay, so we have one question from Tibor. Please go ahead.
Hi, thanks for the overview. So I have one question regarding the building a research workflow, tweaking a few times, rerunning again. SnakeMake has a very nice caching system that automatically, by default, it does not rerun previous rules. So it works perfectly well. But my question goes toward the habits in your research group or community regarding between workflows caching. So it's tagged as an experimental feature in SnakeMake that you can have cache through in your rules. We have been experimenting a little bit and it seems to work well, but it's still tagged as an experimental thing. And I was wondering whether you use it actively or feel a need for caching between different workflows.
It's interesting, we don't use it at all. I'm in the sort of weird, I sometimes feel, I guess this is maybe reverse imposter syndrome or something. I'm not sure that makes sense. I often feel like I am stuck because of the way I teach and because my lab, I bring in people that a lot of my lab consists of people that aren't computationally expert. And then throughout their graduate career, they learn that computational expertise. I often feel like I'm stuck using the first 60% of any software system, because anything advanced means that other people in my lab can't debug it, can't understand it. So I've never used it. And I would almost argue, so okay, so here I'm gonna drop a Kernighan quote on you, which is debugging is twice as hard as programming. So if you write code at the limits of your ability, you won't be able to debug the result. That's a paraphrase. And I really take that to extremes in what I do, like not extremes. I take that pretty far in what I do. I really am in the business of trying to enable my lab to learn, the members of my lab to learn and grow. And I just don't generally have the longevity of people to become expert in things like that. So I do have some experts in my lab who I'm certain use it, but I would not use it in most of my workflows that I'm developing for reuse by people within my lab, because I would worry about the weird behavior that I would find weird if I noticed it, but they wouldn't notice in the first place. So sorry, that's a really unhelpful answer, but that's how I approach these weird advanced features that SnakeMake has. But now that you've told me about it, I will look at it and see if I can write some documentation for it. I'll tell you more about that in a sec.
Thanks.
And then maybe I'll sneak one question in,
Yeah.
then we can go on with the rest of the talk. So you had mentioned on, I forget what slide it was, but you were talking about some of the possible limitations. Yeah, the things you don't love about SnakeMake slide is you mentioned that in the past, SnakeMake has stopped working well past approximately 50,000 jobs. So is this is where you're basically asking SnakeMake to be launching that many jobs for like individual files and things, correct? So
that's right.
Okay, so it's but you could imagine, maybe I'm also trying to think a bit more with my like particle physics workflow hat on. You could imagine that you might only have like 50 to 100 jobs that are then launching like Dask workflows for you that are then scaling out behind the scenes, and that the SnakeMake jobs are much more kind of higher level worker jobs. Is that sort of situation something that you also use, or are you kind of using SnakeMake as kind of both the high level interface for the control as well as then trying to deal with all the distribution?
Right. So what you're talking about should work great. And we use SnakeMake I guess I would say well, let's say in three ways. One is relatively small boutique jobs that are a couple dozen files, a couple hundred files, where a for loop is going to be too fragile, and we don't really care about the automation. We don't really care about anything but the basic automation and failure detection. And SnakeMake works great for that. We use SnakeMake sometimes for large complex DAGs, large complex workflows where large here is like there's probably a total of 20 to 100,000 jobs in the DAG, and that's individual jobs running on individual files, individual collections of files. And that's for some of our papers where we're embracing data reuse. That's we're downloading hundreds of data sets and running dozens of things on each data set. And that's where, past a certain point, that in the past, the DAG solving, the DAG resolver, what's the job I should run next, has sort of has stopped performing well. And so, in fact, then we then move to the third approach, which is basically what you described, which is we sort of black box some of the steps. So we reduce the total number of steps by either writing a shell script that does, 5 of the steps in one. That's relatively rare. Or we as you say, we move to a higher conceptual level and we write a custom thing that does, massively parallel tasks. And when you introduced me, you said something about analyzing petabase scale data sets. We have a public data set that's about 14 terabytes in our sketched version, representing about 20 petabytes of data, and there's 1 million files, and we don't use SnakeMake for that. We use SnakeMake to say, here's a file containing the list of files we want to analyze. Here's some Rust code that's going to operate in massively parallel on those across all of those files. Go ahead and run. But SnakeMake never sees the 1 million files. It just sees the file that contains the list of files, if that makes sense. So I think you've exactly hit on what you do is exactly what we do. That's basically what I'm saying.
Okay, fantastic.
Yeah. And I should say, like, SnakeMake constantly improves. The primary author, Johannes Kuster, is just fantastic and amazing. So that's why I really want to emphasize this is in the past. I don't know if we haven't run into this in the last year or two. So I don't know if that has changed. Okay. So a couple other things. So something that is going to maybe strike people as rather weird is something that we've invested a fair amount of time in. I think I have 4 of these in the lab at the moment, is we write applications around workflow software. And so the idea is, and we actually have a PLOS Comp Bio paper on this, a 10 simple rules paper. And the idea here is that it's relatively easy to embed SnakeMake in a Python CLI. And that means what you can do is you can sort of override the default behavior of SnakeMake with your own preferred behavior and hide a lot of the SnakeMake complexity. So you write a snake file that does some complicated set of things. And then you make it look like a command line application that just has like one entry point, runs a bunch of stuff, and produces a single output file. But underneath, it's actually running SnakeMake. And the upsides of this is that you're basically, you don't have to reinvent job control, failure resumption, scheduling, cluster utilization, all of that. You can actually just pass through a bunch of parameters to SnakeMake from the Python command line. And I have to say it works. It sort of like works at like 80% well. The downsides are that SnakeMake error messages come through regardless, and the SnakeMake error messages will not represent as errors from your application in a very nice way. And then actually the biggest problem I've found is that testing these SnakeMake workflows in general is actually rather challenging. So it's not my 95% solution for writing workflows into an application, but it's like 80%. And I'm happy to talk more about this, but it's just something you should be aware of as an option. And there's this nice description of how to do it if you're at all interested. Okay, so the last big topic that I want to bring forward, and I'd really love your thoughts on teaching this stuff. So many biologists and biomedical data scientists should be using workflow systems, but we have a problem. We don't teach computing or data science to undergrad biologists. In fact, they may actively avoid it. So we have a lot of grad students arrive in grad school with little to no knowledge of computing, and then they're told like, hey, you're going to generate this much data tomorrow. Now you need to learn how to analyze it, and congrats, you're going to be the new expert in the lab. And this is incredibly common. There's sort of this dawning horror maybe overstating, but there's this dawning horror in graduate students. They arrive, they're like, all right, let's do some real biology. And then their advisors are like, look at all the cool data gathering we can do for your system now, for our system now. And then like, wait, I have to analyze this data, but I didn't know that was in my future when I was taking all my undergrad classes. And I would argue that, like, I don't know, again, if this is true in physics, but there's at least more of a culture and community in physics, but we just do a terrible job, I think, in academia of teaching computing basics. You're expected to just pick the stuff up. There is an alternate world where we teach like basic computational lab hygiene, like we teach basic molecular biology hygiene in biology, and we just don't. So often you're stuck making this up in grad school. And I'll say, if you want more motivation here, I'm gonna make an overwrought statement and just say, like, if you look at what biotech companies are doing, biotech companies are generating hundreds to thousands of data sets because it's relatively inexpensive now for whatever disease they're interested in tackling, and they're really challenged in analyzing these data sets. The people that can do the analyses are often computer scientists or physicists that don't know the biology, and the biologists are often the ones producing the data sets and having the questions, and they're usually not able to run the analyses themselves. So I think there's this interesting gap that actually has a lot of societal implications for curing disease, making progress on big health issues. So this is something I feel pretty strongly about. So I've invested a lot of time in it. And my slogan, I'm trying to turn this into a slogan or a catchphrase. Like, it's easy to learn to run one sample, but no one ever teaches you to analyze 100 samples. In bioinformatics, you can always find the commands you need to run by using Google or ChatGPT or something. Like, how do I take this data and get this answer? And then you write that and you run that the command line, and you have some journal of like what you ran, and it's great. And then you're like, okay, but now I need to run this 5 times or 10 times or 100 times and then you need to substitute, sample names in your work, in what you're running. And that's where you start making mistakes. My personal experience is anytime I run something more than 3 times, I make a typo the 4th time or the 5th time. So it's just a completely different kettle of fish for people. So I've been teaching this, and this teaching this has led me to another interesting, I think, realization, which is that I think workflow systems are sort of a cracked mirror of computing. And what I mean by that is every feature in a workflow system exists because of a feature that is lacking in standard approaches to computing. Like most computers don't tell you most execution systems don't tell you when a job fails. They don't give you ways to specify execution flows that aren't linear. They don't give you ways to robustly resume where you left off. Shell scripting languages are lovely, but they're not really designed for robust programming for whatever reason. And that's one of the reasons why things like Python and first Perl and then Python and so on have come along. And then there's something, this is all sort of vague, hand-wavy stuff, but I'll just say, like, right now in computing, there's a pretty hard delineation between single chassis computers and clusters. It's really hard, in my experience, to say, I have a job here, I'd like to migrate it over to that computer over there that has more resources. You need to go through all sorts of rigmarole to do that. And workflow systems sort of can handle that for you with about the same amount of rigmarole, but a lot more efficacy. So another sort of thought on teaching all of this stuff is maybe if we can teach workflow systems well, it's actually a good entry point to understanding how computers work. And so I've been thinking about how to do that. I'm not sure I've made a lot of progress, but I've been thinking about how to bridge from teaching workflow systems to get your work done, which is what researchers want to learn, to then using that as a way to broaden the understanding of how computers work underneath. All right, so with that as an intro, it's easy to learn to run one sample, but no one ever teaches you to analyze 100 samples. And this is maybe teaching workflow systems is maybe a way to teach computing basics. I've been teaching SnakeMake to non-computational people for about 5 years now. And the approach I've taken is actually I just start with shell commands. I say, look, here's a bunch of commands you want to read. You want to download some data. You want to download a reference genome. You want to run something that combines the two things. And you'll notice there's nothing advanced in this. like there's some syntax, but fundamentally you could run these shell commands yourself at the command line and they would work just fine. And you just have to run them in the right order, but that's always going to be the case. So I start with these shell commands and then I tell them, okay, we're going to tell SnakeMake to run the rule download data. We're going to tell SnakeMake to run the rule download genome. We're going to tell SnakeMake to run the rule map reads. We're going to see that map reads fails if you don't download the data first, and then you get a big red error message. And then over the next 2 to 4 weeks, I lead them through adding inputs and outputs, replacing things with templating, reducing redundancy with templating, scaling up to more samples once you have a template. And so we end up with, rules that look that are more connected, like this, where we have, okay, here's this rule, and here are the inputs to this rule, here's the output to the rule, and here's how to use the templating, and so on and so forth. And again, the rationale here is you can always find these shell commands online, but then figuring out how to generalize them to many more data points to many more data sets is the challenge. And so that's what I try and teach. I do this over a period of 10 weeks with 3 different workflows, 3 different common bioinformatics workflows. And I've been told that I think somewhere in the neighborhood of 20% of people go ahead and continue using workflows somewhere after this in their grad work. I'll take it as a partial win, but I'd love to figure out how to make it work. I think we'll make it work better. My understanding right now, my sort of hot take understanding is that I get in my 10-week class, I get people over the logging into a remote Unix system hump, and I get people over the running commands successfully hump, and I get them over the sort of here's what a workflow system looks like hump, but then they don't know how to apply it to their own data. And that's where I am trying to figure out how to do sort of a project-based class next, so that I can take a situation that's not as complicated as their actual research, but that is still a sort of research situation where there's a lot of unknowns and failure. And so they can get them past that first, like, how do I go from knowing what I want to do to actually doing it on my own? So as part of this, I've been writing a draft SnakeMake book. I've been writing a SnakeMake book. There's a draft version that's freely available online. It's open. I don't know if you call it open source. It's openly licensed. The URL is here and on the last slide. And I feel like the intro sections are pretty good. They're based on what I teach. So I have a lot of students that have run through material like this. We start with sort of, what SnakeMake does for you, how to build more useful snake files. This all goes through one workflow. And then I have a bunch of sort of reference level materials for things that are usually like the first set of questions you have after you successfully build a snake file. Like, how do I use wildcards? How do I debug SnakeMake workflows? What are the basic syntax rules? And before you get too excited, I just want to say like half of these chapters on this page are empty. I do think the first like 4 or 5 are pretty robustly filled out, but I'm still very much a work in progress. I tend to work on this mostly when I'm teaching, because that's when I'm highly motivated to give answers to students that aren't just here's a solution to your problem, but a solution to everybody's problem in this space. I've been told by people that this is really a good addition to the default SnakeMake documentation, to the basic SnakeMake documentation. I look forward to your feedback if you are using SnakeMake and want to give this to people. I'll show you the URL again at the end. I will say that I haven't posted publicly about it anywhere because I am not quite ready to take feedback on what I should be doing. So rather than having, at this point, I'm not yet ready for critiques on the structure, but if you have questions that you would like me to answer, I will write documentation for you on how to solve your problems, or at least I'll try. I'll put it in my issue tracker at least. So I am building the SnakeMake book as somebody who programs a lot. So all of my examples are executable and under automated testing. I have continuous integration running. I think there's room for a robust community contribution model once I get the sort of structure, the skeleton basically filled out. And I think it fills an unmet need in the SnakeMake community, in that it's hard to get started with SnakeMake. And, if in the world where I have enough time, I'm not sure exactly where this falls on my list, moment, but I'd really like to build something that can be used for upper level undergrad teaching for people headed for either industry or grad school, and build out lesson examples so teachers don't need to already be experts in order to teach a class. So I'm happy to talk to people that are interested in that. I think that's more or less all I have to say. I'll just put these resources up, and the slides are available in Zoom chat, and I'm happy to I don't know how easy they are to find via Google. I'll say one set of resources that I haven't talked about too much is this Introduction to Remote Computing on the bottom, which is an 11 lecture series with videos for like going from basic shell all the way through to using Conda and SnakeMake and Git, and as well as Amazon Web Services and HPCs. So with that, I'm just going to say thank you. I'm always happy to chat. You can reach me via email on Blue Sky. If you find a relevant GitHub repository and you file an issue there, you might have to tag me in if I'm not watching, but generally I'm watching, and I really like interacting with via GitHub issues, because then the answers are available via Google, which is a big, I don't have to answer it too many times. So with that, I'm just going to say thank you, and I'm happy to take any questions for as long as people are going to hang around.
Thank you very much, Titus. This was a great talk. I've also gone ahead and uploaded Titus's slides to the Indigo agenda, so they're there now as well. Yeah, so we can go ahead and take questions. Please raise your hand in chat. Okay, so I don't see any hands yet, but I have a few, I guess, questions that I would file under sociology, if you will.
Sure,
So,
yeah.
Because I think this is, so for context, Tibor is one of our colleagues at CERN who is the project lead of the RIANA reusable analysis system. Sorry, Tibor, I kind of butchered the full name there. But in which it allows you to use multiple different workflow systems to try and execute a bunch of your jobs at scale on Kubernetes, and SnakeMake is one of them. So he's kind of our HEP SnakeMake expert here. But one of the things that, while we've been trying to use RIANA and the Atlas collaboration and in others, that we've kind of noticed is that teaching people how to use SnakeMake, and also teaching scientists how to make the transition between thinking about, I know how to write my analysis code as a whole bunch of shell scripts glued together with Python and such, and then how to get them to transition to thinking about, now let me re-implement the kind of mental logic to write this as a workflow language, is something that is difficult and seems to also be something that requires some training, or maybe more formal training. As you mentioned, we have a large training effort in particle physics, specifically because, as you already mentioned, at the university level, there isn't this kind of same level of formal training that happens. And so we need to try and get hundreds or thousands of graduate students kind of up to a basic level as well. So I'm interested, so with that incredibly long introduction, I'm interested kind of, what are your thoughts on how in the bioinformatics community you've gone about trying to capture this concept of workflows and have people transition to thinking about workflow languages or workflow engines while they're actually designing their science, their actual, the scientific experiment that they care about doing at the end of the day.
Yeah, so I'm going to say right up front, I don't have any good solutions for you. Sorry. I think that we have, to the extent that we have any advantages in bioinformatics over my understanding of how physics works. And as I said, I've worked with a lot of physicists, but I haven't done much in the way of physics research myself. I think in bioinformatics, we've really embodied, because it's all like data in, data out work. It's all little scripts, programs to transform X to Y. that workflow systems are actually more obvious once you know they exist than they might be in other fields. Just as like as soon as you start analyzing your data, you're confronted with what's that adventure quote, a maze of twisty little passages. You're confronted with, oh my God, I have to install 8 different pieces of software and I have to run each one with a command line that's specific to each of 100 different data files. And so, I don't think, I guess I would say, it is easy to motivate workflow systems at that point, as long as you motivate them through the efficiency from the efficiency perspective. I've never gotten anywhere telling people like, this will make your research, I've never gotten anywhere concrete by telling people this will make your research better, more reproducible, more open, more reusable. Nobody really seems to care about that as their proximal driver. They all acknowledge it, but they care a great deal about this will let you run your analysis quickly and efficiently and make use of all the available compute resources. And that they really respond well to. I think I also, and this is a weird thing to cast as an advantage. I have heard from various people in various subfields of physics, not particle physics, that their fields are very stuck in their ways. There are a bunch of people that know how to do things, and they've known how to do the same thing for 20 years, and they've been really focused on doing that one thing. Certain kinds of fluid dynamics modeling, for example, and there's no way you're going to get them to use a new tool. So a new language, much less a new tool. And so in biology, we have sort of this interesting advantage, and I actually sort of feel okay saying this out loud, although it still feels kind of manipulative. Like every biology professor I've met is more than happy to cede bioinformatics training to me. Like they're like, oh, thank God someone's willing to train my students in bioinformatics. And that gives me a free hand in what to train them in. Like nobody's blinked an eye at me teaching SnakeMake as a workflow system because they don't have the on-ground expertise to second guess me. Maybe that's an advantage, or maybe I've just reached the point in my career where everybody's like, oh, we can't change his behavior, so we just have to live with them. So maybe that's a negative view on the thing. So I guess I would say, like, I think maybe we're in a different space sociologically here. I think it's very hard to convince computer scientists, in my experience, to switch their tools because their tools work well enough for them. Biologists, their tools don't work well for them, so they have to pick up something new. So that's the best answer I can give.
Thanks. That is, I think, a pretty good and useful answer. And I think our community is kind of somewhere in the things are definitely working because we're continuing to get, LHC analyses done, but it's clear that they might be able to be done in a better way. Oh, sorry, go ahead.
So one quick response there. I've heard from some of my colleagues that run HPCs that they're thinking, this is me projecting onto what they've said. So nobody has said this precisely to me, but I think there's an argument from the HPC people that the people, the sysadmins and so on, that using a workflow system is something they not only want to encourage, but are moving towards, making required in some sense, or, as required, at least training in them required, because computer use is so inefficient a lot of the time. So, one of the hooks for things like the software carpentry training, which is something I've been involved in for a while, is that HPCs are increasingly like, you need to take this training before we give you your account. And so that's another way you can do things. You could say, look, we're only going to let you have access to these advanced computing resources or these old clusters over here or whatever, if you use something like SnakeMake, because they decrease our support burden and they increase our ratio of compute efficiency to, time or something like that. So that's just a thought.
Okay, great. There's a question from Gordon, so we'll go to him next, and then if there's time for one more, maybe we can sneak that in. So,
hey, Titus, that was a great talk. Thanks. It's always great to see other perspectives. I have a question, but I just wanted to respond to something you just said. You were talking about, people seem happy to see the training, and you don't know if that's just because, like, they've decided you're set in your ways and you can't be changed. And actually, as a professor who cares a lot about software, but is also very happy to see training to others, it's a combination of two things. One is the amount of time that we have, and the second is that my suspicion is after the students come out from working with you, they're productive. And frankly, from a professor's point of view, that is probably one of the most important things, is how do I get the students spun up doing research efficiently as fast as possible? And
Thanks.
just listening to what Matthew was saying, one of the biggest problems we have is we can teach all these new techniques, but nobody can really do research on their own in particle physics. So you join a team, and inevitably you need to do and use the same software framework, same techniques that the team is using. So the team will contain people that have been doing this for, I don't know, I'm making up a number here, but that's not an unreasonable number, 10 years. So effectively, you've now taught the student all these new techniques, and then you've dragged them backwards in time a decade. So the student can, as they get more confident, right, because new students don't have a lot of confidence usually, they can start to inject new pieces, but it means in particle physics, at least, it really feels like we roll forward slowly. And there's some big breaks, and those are great opportunities where everything, the ground under you disappears and you have to start from scratch. But those are precious few. Okay, on to, and Matthew may want to respond to that, but on to my actual question. So I love your blog, the basement of the ivory tower, or maybe that's
Thank
not
you.
your blog, but whoever is that
It is,
name
yeah.
is. Oh, it is. Okay, the name is fantastic. However, I was trying to look through the SnakeMake book draft and the GitHub repository, mostly because I wanted to see how you did your live documentation, because this is a problem we're currently trying to solve. But both of those links get me 404s.
What?
But the Ivory link works. Um, is I just wondering if this is me, or if somebody else could at, at least. Okay, that works.
Okay, I must have put the wrong.
Okay, so maybe the text is right, but the link behind it is wrong.
Interesting. Okay, sorry. I'll go double check my slides.
No, no, no. I'm sure it's probably me.
So,
Get Yeah,
so I
and the
can
GitHub worked
answer.
as well.
Excellent.
Yep.
So I can just briefly answer you and say, so I started using something called Rustbook. So I've been learning Rust on the side. I don't know if you're familiar with Rust.
Yeah, the
It's a
programming
fun,
language
yeah.
Rust. Yeah.
And I've been using Rustbook, partly because it's blazingly fast, partly because I liked the formatting. And then it also had a robust set of plugins available. But then I wrote my own test infrastructure on top of that goes through and finds the snake files and runs them. And that was rather easy. Um, I'm deeply conflicted about, not deeply, I'm reasonably conflicted about putting a lot of time and effort into writing my own test infrastructure. But as far as I can tell, there's not something that's like, oh, you have snippets of command line. There's not a good test infrastructure for, oh, you have snippets of command line code in your documentation. Please go run that for me. So it is what
Okay.
it is. I mean, that's what
And
Rustbook gave me.
right. And just is there somewhere, do you have an output you compare to make sure that the right thing happened? Is that embedded in the documentation in GitHub?
I actually, we can take this offline. I'm happy to just point you at a couple examples. Because the snake file has Python embedded, I actually include assertions in the snake file about
Okay.
what the results should be. I think that's how I did it. So that's mostly what I do. And then I
Perfect.
stuff the output itself directly in the book. So in some cases, so I'm sure there are improvements, but it's been better than nothing.
Oh,
I
this
guess
is
I would
great.
say.
Thank you so
Yeah.
much.
Thanks. All right. Thank you for the questions and discussion, everybody. It's been great. Thank you.
Yeah, thank you very much for this great talk. It's clearly been very interesting to us. So unless there's any burning final questions, we're 7 past the hour now, so we should probably let Titus go. But again, Titus, thanks very much. And
Thank you.
yeah,